Hello, good morning.
Good morning, sir.
Hello.
Good morning, sir.
Hello, can you hear me?
Yes, sir.
Yes, sir.
Hello. I think,
Yes, sir.
okay. Very good morning. So today I'm going to talk about uh, three small parts of the metabolism, and let me know if it is my slides are visible to you. And just in a second I will show it to you. Yeah. Is it visible?
Yes, sir.
Okay. So today I'm starting with one of the important metabolic cycle or metabolic pathway, which is a gluconeogenesis. The gluconeogenesis refers to the synthesis of glucose from other non-carbohydrate precursors. And basically, you require gluconeogenesis to maintain the level of blood sugar or blood carbohydrate for the brain and muscle, which anytime if there is a metabolic demand, this glucose should be available. Now, this non-carbohydrate precursor of glucose are first converted into pyruvate or enter the pathway at later intermediates such as oxaloacetate or dihydroxyacetone phosphate. So you all know that in glycolysis, glucose is converted into pyruvate. Now this pyruvate has a choice, either it can go into the TCA cycle or it can go to the lactate formation. Lactate formation occurs when there is a oxygen supply which is less than required. That means when you are running, when you are doing extensive exercise, at that time the lactate is being formed and quick glycolysis occurs so that the energy demand is met by whatever the amount of ATP being produced by glycolysis. On the other hand, when there is enough of oxygen, the cell metabolizes pyruvate into carbon dioxide and water through citric acid cycle or TCA cycle, and then you get the complete breakdown of pyruvate, which is started from glucose. On the other hand, this pyruvate, if it is not required for the further metabolism, you don't require energy. On those cases, pyruvate can go back to again synthesize glucose molecule. So that is the process by which glucose can be synthesized from the same product, pyruvate, but you will be learning that it is not the reversal of metabolism. So here we will talk about though pyruvate, which is synthesized from glucose, is again converted back to glucose, but this is happening at the cost of energy. So that is what we will talk about. On the other hand, other than lactate, which temporarily forms from pyruvate, other source of glucose are, for example, amino acids or glycerol. From where do you get amino acid? Amino acids are obtained from the breakdown of proteins. So suppose a person is under extensive starvation and he requires energy. So how does this come? This come from the breakdown of the protein. And that is why you know that even in the patients who are suffering or who is not getting carbohydrate for a long time, they are being given either glucose drip or so glucose drip, they can take care of the need of the brain and muscle whenever it is required. Or if it is not given, what will happen? The protein will break down into amino acid and they will provide the energy. Third source is a glycerol. From where do you get glycerol in the body? Glycerol is the one which is produced from the breakdown of the fat. What is the fat? It is a triacyl glycerol, which is broken down into diacyl glycerol, monocyl glycerol, and further to glycerol. And this glycerol is the one which can synthesize glucose molecule. So you have three major source of non-carbohydrate precursor which can synthesize glucose. Now, the glycerol may also, as you know, glycerol can enter in gluconeogenetic pathway. And how? or it can also enter in glycolytic pathway. So both of them, they are, one is in the forward direction, another is in the backward direction, and this is happening at the point where there is a dihydroxyacetone phosphate. From where you get dihydroxyacetone phosphate? In glycolysis, that you know that fructose 1,6-bisphosphate is present, which is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So that is the point where the glycerol is entering, and these reactions which are making dihydroxyacetone phosphate is given below. That is glycerol. It will be first in presence of glycerol kinase will be converted into glycerol phosphate, glycerol 3-phosphate. This is one of the component. Another is that you have glycerol phosphate dehydrogenase, which is forming dihydroxyacetone phosphate. Remember, it is not in this intermediate. Some of you may wonder why glycerol phosphate doesn't enter. Glycerol phosphate is not the intermediate. You have a glyceraldehyde 3-phosphate. So that is why it is not entering in the metabolic pathway at this point. Dihydroxyacetone phosphate is interconvertible to glyceraldehyde 3-phosphate. So here, once dihydroxyacetone phosphate is formed, this can be converted into glyceraldehyde, and glyceraldehyde 3-phosphate can further metabolize in glycolysis. So this is the way how glycolysis can occur. On the other hand, this dihydroxyacetone phosphate in presence of glyceraldehyde 3-phosphate can synthesize fructose 1,6-bisphosphate by a different reaction. I will talk about those reactions. Now look at this. It's a quite complicated, but if you look at this cycle, which is given on the left-hand side, what you see is that if I look in the forward direction, for a moment, you just don't look at the direction of the reaction. So what I have glucose making glucose 6-phosphate. Though the reaction is different, enzyme is different. Which enzyme is involved here? You will have, in glycolysis, you will have hexokinase. The second enzyme is same of glycolysis. Third enzyme is given different because it's a reversal of the reaction. Third enzyme, you have phosphofructokinase. So you know about hexokinase, you know about phosphofructokinase. These two reactions are replaced by these new enzymes. So phosphofructokinase is replaced by fructose 1,6-bisphosphatase, and hexokinase reaction is reversed by glucose 6 phosphatase. So these two are phosphatases which are removing phosphate group, and you are getting the reversal of the reaction. Why reversal of the reaction doesn't occur in the presence of same enzyme? Because if that happens, then whole cycle will be reversed without any control. So these are controlling reaction. I will remind you that these are the reaction where ATP is being consumed in the forward direction. When glycolysis occurs, these are the reaction, they are irreversible reaction. Why they are irreversible reaction? Because the ATP is involved here. So you require energy for the forward direction. And wherever the energy is required, what will happen at the end of the reaction? It is not 100%, so the reversal of the reaction does not become feasible. Now look at the further reaction. Here, I have shown that glycerol can enter. Glycerol is the one which is being formed from where you get glycerol? It is coming from triacyl glycerol, which is TG, coming from fat. So when fat is, or triacyl glycerol is metabolized, one of the component is glycerol. The other component is a fatty acid. We will talk about how fatty acid is metabolized in the last part of today's lecture. Other steps are common, so I'm not repeating, but what you have is that you have this step, the last step where phosphoenol pyruvate is converted into pyruvate, you have one enzyme. This enzyme converts phosphoenol pyruvate into pyruvate, but here reversal requires two different enzymes. And here I have mentioned that some of the amino acid can enter at this level, oxaloacetate level. Some of the amino acids or lactates, they can enter at the pyruvate level. So amino acid, if they are present in the cell and you require glucose for emergency, they can be synthesized by these routes. So I'm looking at the pathway which is in the backward direction, though it is not the reversal. Why I'm saying it is not the reversal? Because the same set of enzymes, same set of energy cannot replace the whole reaction. Is it clear to you? So in the forward direction, you have a glycolysis. in the backward direction, the one which shown by the arrow in one direction. So you see that step here, conversion of glucose 6-phosphate to glucose, is the one which is not the same. Second reaction is conversion of fructose 1,6-bisphosphate to fructose 6-bisphosphate, is not reversal. Third one is oxaloacetate to phosphoenol pyruvate and pyruvate to oxaloacetate. In fact, if you look at the reaction in the forward direction, this is the reaction. Phosphoenol pyruvate is directly converting to pyruvate. But here you require two-step reaction. Now let us move on. So first in glycolysis, I'll just take the example. Glucose is converting to glucose 6-phosphate by, in the presence of enzyme, which is hexokinase, and this reaction requires ATP. So this is an irreversible reaction. Second irreversible reaction, which I have discussed just now, is fructose 6-phosphate to fructose 1,6-bisphosphate. All of them, they have delta G negative. So to reverse it, it will not be feasible spontaneously. The third one is by pyruvate kinase, which is synthesizing pyruvate. Again, this is requiring, this also is the one where energy is being generated and it has a delta G negative. So overall glycolysis is driven in the forward direction by these three sets of reactions. And these are the reactions which has to be, synthesizing, or which has to be converted into a different direction with the help of another set of enzyme. So I think by this time you are clear that these are the three steps which are irreversible steps, and that is where we are going to talk about. Though in general I have given you how it happens, but there is a different way in which the control occurs. If the control is not proper, what will happen? On one hand, glucose is being converted into pyruvate, and pyruvate will be converted into glucose. So what is the need? Then there is no direction for the reaction, and that's why it is the one which is moving, which is the one which can control the reaction. So let us, when we look at the glycolysis, we look at the conversion from glucose onwards. Here we are looking at the conversion starting from pyruvate to glucose. So let us see what happens at the pyruvate level. So as you have seen, the pyruvate is converting to oxaloacetate first, which is not a common reaction in the glycolysis. So here pyruvate is carboxylated, CO2 group is added in pyruvate, and it is making oxaloacetate. Can you recall this reaction? We have discussed this reaction some time back. This reaction, the one which I'm talking about now, is conversion of pyruvate to oxaloacetate. This reaction we have discussed when we were talking about, when did we discuss about this reaction? Do you remember this reaction?
So in the citric acid cycle.
Yeah, in citric acid cycle, when the intermediate of the cycle were consumed, during that time this reaction occurs, and this is also part of anaplerotic reaction. Anaplerotic means a supplement. If something has been removed, it is going to add on. So it is known as a fill the gap. In Greek, anaplerotic means to fill the gap. So this reaction is a filling of the gap. So it is happening in the mitochondria. So you will be learning that this is again a control mechanism. Whilst all other steps, for example, oxaloacetate to phosphoenol pyruvate, fructose 1,6-bisphosphate to fructose 6-phosphate, or glucose 6-phosphate to glucose, these are happening in cytosol, whereas this is happening in the mitochondria. So you will also learn how pyruvate, which has entered in the mitochondria, comes out of the mitochondria, and then the oxaloacetate is the one which is, which should have, otherwise what will happen? Oxaloacetate plus your acetyl-CoA, they will make the citric acid. But on the other hand, when energy is enough, you require energy for brain and other tissue, this oxaloacetate can be converted into phosphoenol pyruvate. And this reaction happens in the cytosol. So we'll talk about that. The other two reaction is fructose 1,6-bisphosphate to fructose 6-phosphate. This reaction again is controlled by a different enzyme. We have discussed this reaction also when we were talking about how phosphofructose 1,6-bisphosphate, how this phosphatase and phosphofructokinase, they work simultaneously. So this reaction also we have discussed in glycolysis. The last one is the formation of glucose. You will be also entering that the enzyme phosphatase is not present in all the tissue. They are present in only those tissue which can meet the demand of glucose for the metabolism. So then again there is control. In most of the reaction, the gluconeogenesis occurs up to formation of glucose 6-phosphate. And this glucose 6-phosphate, if energy is enough, they will go for glycogen synthesis. That we will talk about in a few moments later. So there are several steps where glucose synthesis is being controlled. Now, this is again elaboration of the last reaction where pyruvate is converted into oxaloacetate. So what happens? There is a carbon dioxide which is added up in pyruvate, and that is what makes oxaloacetate. This is happening with in the presence of ATP and water molecule. So this reaction is an energy consuming process. This will not happen in general if energy demand is there in the cell. If there is a lot of energy present in the cell, then only pyruvate carboxylation occurs to form oxaloacetate. Otherwise, you know that oxaloacetate will be regenerated from the TCA cycle. So in general, oxaloacetate is not formed by pyruvate unless there is a lot of ATP is present, unless you need glucose for the cell. Another reaction which occurs is conversion of oxaloacetate to phosphoenol pyruvate. So again, this is your energy consuming process. So if I look at the energy requirement, when glycolysis is happening, what happens? Phosphoenol pyruvate forms pyruvate and one ATP is being synthesized. Of course, there are two cycles, so I'm just looking at one cycle. One ATP is being produced, but when pyruvate is converted into phosphoenol pyruvate, how many ATP is being consumed? One ATP here and one ATP equivalent here. So almost double the energy is required for conversion of pyruvate to phosphoenol pyruvate, vis-a-vis the amount of energy produced when phosphoenol pyruvate is converted into pyruvate. Am I clear to this point?
Yes, sir.
Okay. So this reaction, to further elaborate, if I look at these two reaction, just to add up these two, you have a pyruvate, oxaloacetate you can cut because one it is produced and then consumed. So at the end, your pyruvate plus ATP plus one molecule of GTP is producing phosphoenol pyruvate. So some of the reaction is that when pyruvate is converting phosphoenol pyruvate, it should be for the benefit of the cell when it is emergency requirement of phosphoenol pyruvate. Otherwise, glycolysis will proceed. Phosphoenol pyruvate will make pyruvate. Now, there is a very tight control in this enzyme, which is a pyruvate carboxylase. What pyruvate carboxylase does? Pyruvate carboxylase forms a oxaloacetate. So this enzyme has two parts. One is N terminal, another is C terminal, and you all know that these two terminal does exist in all the proteins. So at N terminal, it is ATP binding domain. So suppose I have an enzyme here. On one hand, ATP binds. On the other hand, and the C terminal, it's a biotin binding domain. So biotin binds over there. So biotin is, what is a biotin? Can you recall? Biotin is one of the vitamin which is involved in C group transfer of carbon dioxide. So here the enzyme phosphoenol pyruvate, they do have, the enzyme, this carboxylase enzyme, they do have a requirement for the biotin. Why biotin is required, that you will be learning in slowly when you go in deeper in the biochemistry, but it is the one which can act as a arm. Arm means hand, your hand. It picks up CO2 from one side and gives it to the another side. So it's a swinging arm mechanism. That means it picks up one substrate. So it's a two substrate which is present on the enzyme, one on the N terminal, another in C terminal. It picks up one of the C terminal thing to N terminal, and then the reaction occurs. So this is again the same thing. How it happens, the ATP in presence of bicarbonate, it is making a intermediate, and this reacts first with biotin enzyme complex, and CO2 is formed. So this enzyme, this is known as carboxylase enzyme. What it will do is that it will have CO2, which is bound to the biotin. And you know that biotin is nothing but a vitamin which has been converted into its coenzymic form. So what will do this biotin? It will take carbon dioxide, make it approachable to pyruvate. Once pyruvate and carbon dioxide come close to each other in the presence of enzyme, you get oxaloacetate. So this is a mechanism how this happens. The delta G, so CO2 is attached to the biotin and it is quite activated. So it is activated carbon dioxide. This is also known as activated carbon dioxide. So this activated carbon dioxide is the one which is responsible for synthesis of your oxaloacetate. Now, there is an allostatic mechanism. You remember that what is allostatic is that the binding of one of the substrate activates the enzymatic performance. There's further detail because this is a very important enzyme. People, they need to know how this happens and how oxaloacetate is shuttled into the cytosol and converted into phosphoenol pyruvate. Why I'm talking about is that as you learned that oxaloacetate is synthesized inside the mitochondria, and it has to work. If it has to synthesize, pyruvate, it has to come out of the mitochondria, then the reaction will occur. So these steps which are written here, I will try to summarize in the next slide. So what happens here? There is a mitochondria. Your pyruvate initially is synthesized inside the cytosol. They enter in the mitochondria, but once it enters in the mitochondria, pyruvate is either making acetyl-CoA and of course going for TCA cycle. Alternate is that if acetyl-CoA is good enough, there is a control which you have learned in the TCA cycle that acetyl-CoA can be formed only when you have energy requirement. Otherwise, you have several metabolites which can control the reaction. So pyruvate can synthesize oxaloacetate. This can also be synthesized when there is an aplerotic demand for the synthesis of oxaloacetate for running of the TCA cycle. Now, once oxaloacetate is utilized by the TCA cycle, there is no problem. But if it is in excess, what will happen? They will be converted into malate. Look at this. This is happening in presence of another enzyme, and it requires NADH. So whatever NADH is in excess, so this reaction will happen when you have excess of NADH. When will you have excess of NADH? When there is a lot of energy in the cell. So that is the way malate is formed. Now malate will move out of the mitochondria, and again this will regenerate NADH. So actually NADH, which was taken out from the mitochondria, that comes out in the form of your oxaloacetate converted to malate, and malate is again reconverted into oxaloacetate. So these two are again two different enzymes, and that's the way the oxaloacetate synthesis occurs. So once oxaloacetate has come out, now the reactions are reversible. I'm just going back to the first reaction. So here, what I have shown is that this oxaloacetate has come out. Now, second reaction, which is a reversible reaction, is happening in presence of phosphoenol pyruvate carboxykinase. This enzyme is present in cytosol, and they can synthesize phosphoenol pyruvate. Look at this. This is again happening at the cost of energy. So what I'm trying to say here is that conversion of pyruvate to phosphoenol pyruvate is energy costing process, and this will not happen as long as energy demand is not there, and unless there is a lot of energy present in the cell. So once phosphoenol pyruvate is formed, you look at all these reactions are reversible up to here, fructose 1,6-bisphosphate. After that, these two reactions I'm going to talk about. Okay, so next reaction is fructose 1,6-bisphosphate to fructose 6-phosphate. Again, as earlier we have discussed in the TCA cycle, that it is again controlled by a specific reaction. Can you recall how it is being controlled? When fructose 2,6-bisphosphate is formed, that acts as an allostatic regulator that activates phosphofructokinase. Otherwise, this reaction can occur. So fructose 2,6-bisphosphate, if that is present, they can activate the reversal of this reaction. That's conversion of fructose 6-phosphate to this. If that is not there, fructose 1,6-bisphosphate can be converted into fructose 6-phosphate. So this is allostatic control which is happening. Now next is generation of free glucose. This is again very important. Glucose 6-phosphate, once it is synthesized, so far from fructose 6-phosphate is synthesized. From fructose 6-phosphate, glucose 6-phosphate conversion is not a problem. It is a reversible reaction, it happens. In most of the tissue, the formation of glucose 6-phosphate is the end step of the your gluconeogenesis. But what happens if you require glucose? It has to be in the form of glucose. Now you may have asked question, why glucose 6-phosphate is the end product of most of the cellular gluconeogenesis. The reason is that glucose 6-phosphate is phosphate rich. It is energy rich as such, and it can be converted into glycogen. It will not require activation, or it will not require phosphorylation. And that is why in most of the cell, glucose 6-phosphate is, at this point, the reaction stops, and only in very few cells, the reaction of conversion of glucose 6-phosphate to glucose occurs. How it happens? So there is a controlled reaction. The controlled reaction happens if the enzyme is present. So most of the cells, they do not have glucose 6-phosphatase, the enzyme which regulates conversion of glucose 6-phosphate to glucose. The second, that the enzyme present only tissues whose metabolic duty is to maintain blood glucose level. And that is why it happens in liver and to very less extent in the kidney. So what happens again, even in the liver cell or kidney cell, there is a very interesting thing which happens. In these cell, you require a lot of enzyme to convert glucose 6-phosphate to glucose. So this is a very simple reaction, but requires several steps. Here, you can see glucose first enters in the endoplasmic reticulum, and in the endoplasmic reticulum membrane, you have an enzyme, glucose 6-phosphatase. So what will happen? Glucose 6-phosphate enters in the endoplasmic reticulum. It is being hydrolyzed, presence of glucose 6-phosphatase. This enzyme requires a calcium binding protein, which is SP here. Once it is broken down into PI and glucose, these two are transported outside the endoplasmic reticulum by two more transporters. So how many proteins are involved? One protein, calcium binding protein. Second, enzyme itself. Two. Third is a T1 which transport. Fourth is the one T2 which transports PI. And this one is the one which transports glucose. So look at the mechanism, the amount of these transporters, the entry inside this endoplasmic reticulum. Everything is controlling that glucose, how glucose should be synthesized, because once glucose is synthesized, then it will be utilized in the cell. So to sum up, I think this is the last slide for this gluconeogenesis, and it also summarizes how gluconeogenesis is controlled and how glycolysis occurs. So on the left hand side, you see the reaction. So glucose to fructose 6-phosphate, uh, no issue, and then fructose 6-phosphate to fructose 1,6-bisphosphatase. This is happening in glycolysis, and you look at the control. Fructose 2,6-bisphosphate controls AMP; these two activate, whereas ATP, citrate, and proton, they deactivate the process. So you see that these processes are tightly controlled. On the other hand, these sets of the control, they activate here. So suppose citrate is inhibiting, citrate will activate. AMP is activating, AMP will inhibit here. Fructose 2,6-bisphosphate is activating here, they will inhibit here. So you see, on one hand, plus shows the activation, minus shows inhibition. So citrate, which was inhibiting this phosphofructokinase, will activate this process. So if citrate is more, that means more energy is present in the cell, and then it can go for gluconeogenesis. So fructose 1,6-bisphosphatase 2-phosphoenol pyruvate is formed. Then in forward direction, that means for glycolysis, pyruvate kinase works. It is fructose 1,6-bisphosphate, and ATP as well as alanine are involved in the controlling them. Some of them negative controls, one of them is a positive control. On the other hand, these two steps can make phosphoenol pyruvate. So this whole thesis shows you that there is a reciprocal regulation of gluconeogenesis and glycolysis. The one which activates glycolysis will inhibit gluconeogenesis. The one which activates gluconeogenesis will inhibit glycolysis. So I'll stop at this point. And you have also seen, if I calculate the cost of energy, it requires more energy for conversion of pyruvate to glucose than glucose to pyruvate, the amount of energy being produced. And that you have seen on these steps, on the last terminal steps. So any questions so far? If there are no questions, we will move on to the next part, which is how glycogen is metabolized. Now, you have learned that glucose 6-phosphate is formed, and glucose 6-phosphate, once formed, they can enter in the glycogen synthesis. On the other hand, glycogen metabolism we are learning. Why glycogen metabolism? Because glycogen metabolism is the major source of quick energy. Glycogen is the only one which is stored in the form of carbohydrate in most of the animal cells. So first, very quickly, we will talk about how glycogen is being metabolized. So if glycogen is there, every time one glucose molecule is being released, so rest will be glycogen n minus 1.